Okay, this is still morning. So good morning, everyone. Today I'm very excited to present this BE3D, a computational workflow designed to interpret structure and function relationship for base editor tiling screening. Okay. So quick overview of today's exercise. I'm going to start with the background on base editor tiling screening. And then I'm going to explain why we need a computational method on it. Then I will introduce BE3D, our pipeline, and then some results highlighting on 2 case studies. So base editing, or shortly BE, is built on a CRISPR gene editing system. But unlike the conventional CRISPR editing, this base editing can sophisticatedly introduce single mutation or single point mutation by chemically coding those amine or amino group from DNA bases. And this tiling mutagenesis is ideally introduce a lot of mutation across a gene or a protein. So by combining these benefits of the precision of base editing with the resolutions of this mutagenesis, we can systematically introduce point mutation to study protein function at high level and high resolution. So I brought an example. So to do the base editor tiling screening, what we need to do is starting with designing the guide RNA. So guide RNA here is sgRNAs, which guide or direct base editor to the target site on the DNA. So we covered a lot of guide RNA on this gene or protein, and then transduced to the cells. And what we need to do after that is measuring the abundance of the guide RNA in cells. So we actually calculate log-fold changes between this control group versus those cells with the mutations. And then we can tell whether these effects or mutational effects on protein function or drug resistancy or sensitivity, or also we can say the cell viability. So I would say that this base editor tiling screening is ideal for assessing the protein domain essentiality as well as structure function mechanisms. So 2 case studies here. The first one is about DNMT3A, which is DNA matrix transferase, and important for this chromatin remodeling and then development. So our collaborator, Brian Liu's group at Harvard, conducted base editor tiling screening on DNMT3A to characterize what mutations or what residues are important for this DNMT3A function. The other case study is on MEN1 or mannin protein, which is a chromatin adaptive protein and important for assembling and stabilizing these chromatin regulatory complexes. And there was a study saying that acute leukemia is quite important, and then this mannin-1 inhibitor is the target for the leukemia drug. And they found that mutations happening in mannin-1 is important to know the drug resistancy. So this study actually conducted base editor tiling screening on 2 different cell lines on this mannin-1 protein. There are definitely challenges and limitations of base editor screening. So first one is coverage of these editors. So I brought in here the PAM is protospacer adjacent motif, where this base editor can recognize it on DNA and then bind to the target DNA. So without this PAM sequence, such as here NGG, base editor cannot recognize the target or target sites. So we need this kind of PAM sequence to modify it, as well as base editor is cutting off the amine group from the bases. We cannot get all the 20 different types of outcomes of the mutations. So there are definitely coverage limitations. And then if you look at the another important thing is efficiency of the base editors. I here brought in one example, which is ApoBeg1, the cytidine base editor, which converts C to T. But this cytidine base editor has a nucleic acid preference. So C after the T is preferred over the C or after the G. So with that, if we have the signals kind of LFC with this different preference, we get the low signal from those C over the G. So that will mislead us to get high first negative interpretation of biological outcomes. And then there will be definitely efficiency difference between cell lines and species. And the last part of about if we have a multiple screens conducted base screen, then how we can get the consensus research across those variability of the cell line and then base editors. So that's why we need a systematic computational workflow from those conventional base editor tiling screening pipeline. Hopefully we can actually tackle those aforementioned limitations and also allow to interpret higher level of the structure function relationship with this pipeline. So this pipeline consists of the 3 steps, but the most important hypothesis on this pipeline is if there are functionally important residues, but they're not could be targeted by base editor, but they can be still inferred through 3D normalization and clustering of those nearby high signal residues, they are edited by base editor. So we start with the QA, which is quality assessment of each screen, and then we just pass only QA pass screens to the next step. From the next step, what we do is calculating a new score, which is called LFC3D, a 3D aware LFC value based on 3D normalization. I'll introduce a bit more detail later slides. Then the next part is metaclass 3D. When we have multiple screens from the base screen, how we can aggregate them and interpret and get a right consensus research on this. So the QA is how we assess the quality of the screen. So we brought here the biological hypothesis, which means when we treat guide RNA into the cell, we could get roughly 6 different mutations. No mutation silent to splice and nonsense. So red 2 mutations are very deleterious if we have them on a cell. And the first 2 are milder effect mutation, we can think. So the biological hypothesis here is when we check the distributions of those LFC between deleterious versus this mild effect mutation, they will have very different. Such as here, example, DNMT3A base tyrant screening. You can see in the middle, they have mild effects by having median value of almost 0, but those case mutations have LFC median value of 1. So they are quite different. And that is a kind of example of good screen. And we need to quantify to evenly compare across the screen. So we use 2 statistical methods. Here is a Mann-Whitney, which can calculate the distance between the distributions of distance. And also the Kolmogorov-Smirnov test is telling how this distribution have a different shapes. So with that, we can actually check whether the screen passed the QA or not. And the second part is, if you remember, we have a lot of guide RNA to cover the whole gene. So a lot of guide RNA can hit one position with either same mutation or different mutations. But somehow we need to pick just one readout or LFC value per position to map on the 3D structure. So here is how we prioritize those values, but we are showing averaging them. So on this 719, we averaging those experimental value and assign 0.001. And then, but users can actually pick this platform, allows the user to pick by either average or minimum or maximum across these different LFC values. So now we have the LFC value per residue, then how we can map on the 3D structure. So there are many structures available if you have very famous protein. Here we brought an example. So we have developed a genomics 2 proteins portal like last year from our group. And then we use this one for giving an idea that saying how many structures, 3D structures are available there, and also AlphaFold structure. And then actually users can pick which structure could be more suitable for based on what base editor tiling screen conducted. So if your base editor tiling screen conducted on a specific inhibitor bounded, then you probably need to go with that PDB structure. But as a default, we go with AlphaFold structure, which has the most extensive coverage across the protein without missing residues. After that, the important part is actually calculating the score called LFC3D. The basic hypothesis is saying that functionally important residues should be nearby in 3D space, here the 3D structure. So for that, we search the 3D neighbor residues by drawing the 6 radius angstrom and then check their LFC values, and then we average those LFC values and then assign to each position. But users can actually tweak these 2 parameters, which is the radius or also it's a function. So not just the averaging, but that can sum or pick a min or max for that calculating this score. So I here brought 2 results. The first part, the top is about experimental results. You can see that from the DNMT3A case study, the experimenter got 127 LFC value or readout from experiment. And then 40 out of them are actually meaningful signal or hits. You can see in the blue. How we can identify these blue signals? We use conventionally calculate z-score calculation, z-score, and then that's actually telling how these LFC from missense variants are significantly different from those distribution of control. So control means is there missense, no or silent mutations. So we draw the 5 percentile, the top 5 percentile significant LFC of the missense as a hit. And at the bottom scatter plot is showing that if we do the LFC 3D calculation, we got 3.1-fold increase of the signals, showing 399, and as well as 3.4 increase of those blue scatters. So saying that we have huge increase of the signal as well as those hits. But important message from here is we are not introducing or we are keeping this noise and hit ratio as this from the earlier experiments. And here I want to highlight just one thing is when we first designed the guide RNA, we can actually screen the PAM sequence available. So from here, the DNMT3A, we can only hit 264 residues out of 912. But as you just saw that, experiment only got a signal on 127, which is the half of the base editor efficiency. But most important part from here is those regions in magenta. And then you can see that 65 and 68% of those regions are coming from not editable regions by CBE. So LFC3D actually very useful, not only for those regions have very low base editor efficiency, but also can be used to get a signal for those non-editable regions by CBE editors. We got a lot of hits, but still this coverage is 10 to 20%. And then that hits are quite sparse across the 3D protein. So we need a somehow good way or better way to interpret them. To do that, we map these hits on the 3D structure and then do the 3D clustering or spatial clustering and remap it back onto the 3D structure with our platform called Genomics 2 Protein Portal. So you're seeing the Genomics 2 Protein Portal with that BE3D result, the hit clusters, but this is actually coming from experiments. So basic experiments highlight important regions in DNMT3A first, cofactor binding site, and then dimerization between DNMT3A, DNMT3A. But here you can see that these regions are not highlighted by one color, but more than 3 or 4 different hit clusters. This dimerization site has not covered 100% by hits from experiment. So we can see that a bit limited coverage of the experiments. However, if we ran on the LFC3D, it's highlighting a whole cofactor, not 100%, but see extended coverage on this cofactor or inhibitor binding site, as well as this dimerization interface is now fully covered by LFC3D scores. And interestingly, when you actually align these hits with clinically verified pathogenic variants from CleanVar database, we saw that those boxes are well-aligned hits and pathogenic variant. The 60% of them are coming from not targetable regions by CBE editors. So this is actually signifying our pipeline can be used for potentially telling or identifying the pathogenic variants as well. So the last part of the B cluster is about meta aggregation. When we have multiple screens and how we can aggregate and get a consensus signals, we picked the summation as an aggregation function as a default, but users can pick just any function within there to get the hits across the multiple screens. Here I brought a MEN1 example. So the partner group conducted base editor on the mannan protein on 2 different cells. And the important thing here I want to highlight is we capture, so LFC3D, we capture very key residue that gave like drug resistancy, which is highlighted in orange, but they were not identified from real experiments. When we did the meta aggregation on the right side, what you can clearly see from here between these two will be this empty space. So when we did the meta aggregation on the 2 different screens, we reduced the noises from NTD domain, but we are keeping the important signal where the drug resistance is happening. But also we can see that a bit of potential drug resistance site, they are quite well aligned with ligand binding site. You can see here, as well as those predicted pocket sites. So we're still in preparing the manuscript of this pipeline, but soon to be ready for also the GitHub and Colab, so anyone can run on the Colab. And this should be available, a map on the available G2P portal, so users can actually map those hit clusters and then align with a lot of information from this genomics to protein portal to interpret the structure function analysis. With that, I hope I convinced that this BE3D pipeline can streamline the conventional base editor analysis by showing this increase the coverage over that non-editable regions, and also meta class show how we can deal with the meta, a lot of screens at getting a consensus research. And then also the 3D clustering or spatial clustering is really good to inferring that high order structure function relationship. With that, I'd like to thank first my supervisor, Sumaya, for all the advices and then help, and Calvin and Surya for the implementation and development of the pipeline, and lab members and then collaborators this is here, and the funding bodies as well. And then thank you for your attention. Thank you.
Thank you for the talk. Are there like any questions? I will get to then kickstart with one. First of all, like a general question. Do you know, like, if from like a molecular point of view, it is known like where like the variation efficiency come from, especially for GC?
So why we are getting the variant efficiency?
Yeah.
Yeah, actually, so I don't know fully, but what I know is, so T has a small one ring, 6-member ring, and G has 5 and 6-member ring. So G has a bigger than the thymine. So the base editor can easily targeting thymine over than G. And there is another biochemical evidence that I think the T has a more hydrogen bond and vice versa. So the biochemical and the structure size-wise, thymine is more preferred over the G. Yeah.
Okay, thank you. Any other questions? Okay, I will have like a second one then. Because like you're adopting like this quantile approach for your selection, are you also like applying some false discovery rate control procedure?
So quantile means?
Like in your procedure, right? You fit like a threshold at the 5% quantile, right?
Oh, yes.
But then do you control somehow like the false discovery rate?
Oh, so yes, that's a really good point. So here we are only showing that 5% high, right? But when we actually do the meta cluster 3D, which aggregates signals across the screen, that we get augmented signal across the screen, then we can actually put more stringent threshold, like a 1 or 1.5% to get the more meaningful signals. Otherwise, we get a bit more noises by accumulating. So yes, we can adjust this threshold, and users actually can adjust this signal to get only meaningful signals from their interpretations as well. Yeah.
Wonderful, thank you. So if there are no other questions, I guess we can thank the speaker again.
Thank you.